Hi, I'm Mia and I've spent the last 8 weeks doing a project in Mark Banfield's lab at the John Innes Centre exploring plant immunity. I've always been fascinated by plants, in particular how a knowledge of plant sciences can help us with the challenge of producing crops to feed a rapidly growing population. It was because of my interest in this, as well as my want to get experience in a lab, that I applied for this summer project. So my project has been specifically based around plant pathogen interactions. So pathogens produce effector proteins which enter plant cells, and inside the plant cells there are receptor proteins. And those receptors, if they're able to interact with the effectors, can activate the immune system of the plant. And if the immune system is activated, the plant can protect itself from infection. So the specific project that I've been doing is trying to characterize which effectors are able to be recognized by a specific receptor. And this is important because if we're able to understand effector-receptor interactions more, then we might be able to engineer receptor proteins to be able to recognize more effectors. And in doing this, we might be able to produce plants which are protected from more infections, such as wheat blast and rice blast, which are major threats to global agriculture. So I've learnt a range of techniques during this project. One of the main ones has been agrobacterium-mediated transfection, and this is putting the genes of the receptor and the effector protein into bacteria, and then using those bacteria to go into plant leaves and express the proteins in plant leaves. Those plants are then grown in a controlled environment, and a cell death interaction seen as spots on the leaf shows that a receptor can recognize a specific effector. I really enjoyed my experience of doing a project in the lab over this summer. It showed me how exciting and interesting working in a lab can be, and it's definitely making me consider the possibility of doing a PhD after I graduate from my undergrad. I want to say a really big thank you to the BSPP who funded me to be able to do this project, as well as everyone in the Banfield lab for all of the masses of help they've given me over the project.